Welcome to all those near and far. I am Dr. Renee Xavier and a DARPA Innovation Fellow. Welcome to this workshop for the Advanced Research Concept Discovering Unknome Function, or DUF. In this workshop, I will share more details on the Innovation Fellowship and the DUF ARC opportunity, and you will hear from experts in this diverse area of research. This newest ARC opportunity aims to rapidly generate high-confidence gene function annotations for breakthrough discoveries in biotechnology, specifically focusing on those parts of the genomes that are still unknown, the unknome. The biological treasure that is waiting to be discovered in the unknown could lead to game-changing technologies in multiple research areas. One famous example is the discovery of Taq polymerase in 1976 that revolutionized PCR. Taq polymerase is a thermostable enzyme isolated from a bacteria that resides in deep-sea hydrothermal vents. The functional annotation of this single enzyme laid the foundation of sequencing technologies as we know them. But if the unknown is such a treasure trove, why does it exist? Why are certain genes so hard to annotate? To begin to understand this challenge, I interviewed many scientists, some of whom you are going to hear from today, and then used the 6 Sigma 5 Whys exercise to identify the root cause of the problem and ultimately develop this advanced research concept funding opportunity. The speakers today are going to cover the current challenges in this field in more detail. Overcoming these challenges could shrink the unknown, like the complete annotation of the biotech workhorse E. coli, of which 35% of its genome still remains a mystery. Right now, research is at a technological sweet spot. The last decade alone has seen great advances in nucleic acid sequencing, single-cell research, mass spec instruments, and machine learning. Now is the time to pool these resources and focus on high-throughput discovery of unknown function. The DUF ARC opportunity has 2 technical challenges. 1, high-throughput prediction, and 2, validation of gene function. Today's speakers are going to talk in more detail about the current methods they use in their research and the current challenges they face. And while you're listening, I want you to brainstorm ideas for designing an experiment to address these challenges in your own area of research, which could possibly lead you to submit an abstract to the DUF funding opportunity. And I'm now going to give you some guidelines for designing a robust experimental design, so take notes. In general, a successful abstract will be innovative and outline how your idea is better than current state-of-the-art. There needs to be a clear research plan explaining the experimental design, discussed on the previous slide, and goals of the study. Finally, you must have the technical abilities to perform this work in both equipment and trained scientists. This would be a great opportunity for a postdoc to generate preliminary data. The overarching goal of the DUF Advanced Research Concept is to cast a wide net and capture a diverse set of innovative ideas to break the technology barrier that is creating the bottleneck in gene function discovery. Breaking this barrier will result in high-confidence functional annotation in days, not decades, which would be a game changer for many areas of biological research. Please submit all DUF-related questions to duff@darpa.mil. Thank you for your participation, and I look forward to reading your innovative solutions to shrink the unknown. Dr. Anne Rashandra Carvanas is an associate professor in computational and systems biology at the University of Pittsburgh School of Medicine. Her lab focuses on the molecular mechanisms of change and innovation in evolution. Her presentation is entitled The Meanings of Function in Biology.
Hello. What is biological function? This is actually a very complex question because the word function in biology has been very debated and has been the subject of much literature. A good example of this is the so-called ENCODE controversy. In 2012, the ENCODE Project Consortium published their flagship article, An Integrated Encyclopedia of DNA Elements in the Human Genome, whereby they summarize the results of many experiments measuring different types of biochemical activity, such as transcription, histone positioning, across the human genome. And because they found reproducible biochemical activity in most of the genome, they summarize their finding as in 80% of the human genome is functional. This claim proved to be extremely controversial, and in particular with evolutionary biologists, with perhaps the best example of a controversial article following this ENCODE publication is this one from Dan Grauer and colleagues, entitled On the Immortality of Television Sets, Function in the Human Genome According to the Evolution-Free Gospel of ENCODE. While this particular article is particularly scathing in style, the sentiment that it expressed was shared among many evolutionary biologists, because the idea that 80% of the human genome is functional was seen as flying in the face of 100 years of evolutionary theory, because most or all actually estimates based on evolutionary conservation suggest that less than 10% of the human genome is conserved through purifying selection. And for evolutionary biologists in general, conservation is function, not biochemical activity. Many other articles have been written expressing their opinion on this controversy, and at the end, it really boiled down to what is function. Perhaps one of the most thoughtful articles was authored by some of the ENCODE authors themselves in 2014, simply suggesting that there are different empirical lenses into function. This is not a sterile wording debate. It really does matter how we look empirically at the genome to understand how it works and conclude what is functional or not, with, for example, some overlap between what is being conserved and what is being protein coding, for example, but not a full overlap. Same thing for the evidence for genetic perturbations delivering phenotypes or chemical activity. These different lenses are not equivalent to each other. They all give different information, and they cannot all be summarized in one word, function. It really matters in the practical science enterprise what we mean by function, because there are many consequences, like funding or picking candidates for studying new genetic elements. Only the functional ones are interesting, but what are the functional ones? This is particularly interesting to me for my personal research, because I study the evolutionary origins of new genes through the process of so-called de novo emergence, whereby new protein coding genes can evolve from previously non-genic sequences that become translated. The new genetic elements that are newly translated, which I refer to as protogenes, are well expressed by definition and can contribute to cell and organismal biology, but they're only found in one species or lineage. They are not conserved, and they certainly, because they are novel, cannot have a function in the classic evolutionary sense of a reason for having been selected for millions and millions of years. when is a novel gene functional? This question was really gnawing at me because of the complexity of how to think about function. So I decided to collaborate with a philosopher, a biochemist, and a rhetoric scholar in an article we published a few years ago entitled The Meanings of Function in Biology and the Problematic Case of De Novo Gene Emergence. The four of us collected 20 abstracts in the field of de novo gene births, chosen to represent different organisms, different methodologies, different country of origins. And we simply read all these abstracts and tried to see what may have the author meant when they wrote function or functional in their conclusions of whether a potential new gene was functional or not. Discussing what we were reading about, we came up with exactly 5 meanings that we thought the authors meant. Either they were describing the evolutionary implications of the gene, or the physiological implications, or at a more molecular level, what interaction the expression product of the gene could be having with other molecules in the cell, even what were its intrinsic physical capacities, such as the presence of specific protein domains, or expression, such as tissue-specific expression, for instance. Then in some cases, we didn't know. It was really too vague for us to imagine. But now, armed with those 5 meanings, this sort of dictionary for function, we then went and read individually the abstracts again and tried to each assign 1 meaning for each instance of function over these 20 abstracts. And we found out that we completely interpreted the different sentences differently. The graph here on the left shows that for how many instances of the word function, functional, did we find 1, 2, or 3 or more meanings. And you can see that there were only 5 for which the 4 readers, the 4 of us, agreed on 1 meaning that was also the authors meant. And otherwise, we always disagreed. So that shows that without specifying what function means or qualifying it, giving it context to help the reader, your readers will interpret your goals or your conclusions differently. Then the 4 of us then discussed and basically came to a consensus by convincing each other. And in this consensus, then most instances of function we found to have only 1 meaning, but often still 2 or 3. The most often that we found was physiological implication and evolutionary implication, the most high-level definitions of function. So given this complexity that function can mean and has been written about to mean, and how readers and scientists can think about and interpret conclusions differently, in my lab, and that would be a dream if this was extended beyond in this project, I strive to consider the different components of function independently of their relationship to each other and their relationship to each other. So like for a given locus, when and how is it expressed? What are its capacities? For example, where does it go in the cell? How does it interact with the system? What are the implications of these interactions? And how does that influence the evolutionary fate of a locus? Whether it's going to turn over, change, or become retained. Thank you very much for your attention.
Dr. Olayton is a systems analyst at the Jackson Laboratory for Genomic Medicine in Connecticut. His work focuses on supporting and training biologists and scientists in the development of computational tools and the analysis of next-generation sequencing datasets. He has conducted gene expression studies on complex phenotypes and neurological disorders. He is interested in developing reproducible bioinformatics pipelines. With over a decade of experience in research computing, he is passionate about African bioinformatics, reproducible research, and capacity building, and he organizes webinars, codeathons, and workshops in order to train life scientists from more than 44 countries in genomic data science skills.
Hi, everyone. Thank you for inviting me to this workshop on discovering unknown function advanced research concept. My name is Olaitan Awe, and I'll be talking about building reproducible pipelines. So I would like to start by defining what reproducible code is. And my definition is that a code or set of codes that you build into a pipeline is reproducible if the result of an analysis does not depend on the specific computational environment in which data processing and analysis originally took place. So the workflow will produce the same result when we run on different computing platforms. Then I have a framework that I've defined for reproducible code, which basically starts from collecting data and then developing the pipeline or codes. And then when you run your pipeline against the data set, then it generates some output, which you then need to interpret. For omic data science, which is my specialty and focus, that framework will translate into collecting data, which is biomedical data or omic sequences, for example, and then you curate the data. You develop the pipeline or code, and then you interpret the output and present the data in order to advance the understanding of biology and health. Then I also have some steps recommended for writing code and ensuring that the code is published in a findable repository like on GitHub. So when you have your GitHub repository where you want to build out your pipeline, I have some recommendations in terms of the structure of the GitHub repository or which files should be there, just to guide you on building your pipeline into a reproducible pipeline. I would recommend having a data directory where you can put, for instance, things like your data generated as part of your analysis. Have an accessions directory where you can also include things like the SRA or GEO or ENA or RefSeq or GenBack accessions, so that if someone wants to reproduce the work, they know where you got the data from in terms of public repositories, and they know the specific accessions to pull down. And then when you generate figures from your analysis, you can put them in the figures directory. Then you have a scripts directory where you can put your code for instance, dumping data off of maybe the sequence with archive, code for doing your maybe alignment, or building out your reference genome or transcriptome, depending on the type of problem you're trying to solve, and different type of things, maybe code for doing your variant calling or other types of steps that you have in your analysis. You can have a directory for docs where you can include things like documentation relating to the pipeline, or sometimes even papers that you read as part of that project that you're working on. Then have a directory for output where you can put things like data generated from running the workflow. Have a workflow diagram, which is basically a step-by-step sequence of tasks that need to be done in order for the pipeline to be completed. Have a notebooks, Jupyter notebook or R notebook or Markdown that you can use for demonstration, so that anyone who picks up that repository can actually, kind of run through that notebook to see, how to run that pipeline, have an example data set also that they can work with. I would recommend the license to be, open license, for instance, like MIT, and have a README file that can also guide the user in understanding the objective for the work, the background, and potentially the contributors to the pipeline and things like that. In terms of workflow management systems, one thing I would like to say here is that they enable reproducible coding. And examples of those frameworks include Nextflow. Nextflow is a pipelining framework that allows bioinformaticians to build workflows that can be rerun in different computing environments, and you can reproduce the results. It has lots of benefits like interoperability, component reuse, reentrancy, parallelization, and it allows for the use of containers. And you have other workflow management systems like SnakeMake, which is basically based on Python. You have Cromwell, and you have Galaxy. So any of these can be used in building out your pipeline. One thing I would like to encourage is to automate your pipelines as much as possible. So you can use different scripting languages that you're comfortable with, maybe Bash or Python or Perl or Java or C or C++ and any other one. But as much as possible, repetitive tasks should be automated. There are some life science project categories that I would also like to highlight quickly for those that are quite interested in bioinformatics and computational biology. So you have bulk transcriptomics, metagenomics, human genomic variation. Pipeline development is also a category in the life sciences that, one could decide to focus on. Biomarker discovery, cheminformatics, clinical applications, drug and vaccine design, antimicrobial resistance, population genomics, genome-wide association studies, polygenic risk scores, Mendelian randomization, structural bioinformatics, software development, epigenomics, oncology, plant genomics, machine learning. So as we do all these, projects in trying to find unknown functions, we can delve into any of these life science categories in our various projects. You want to start writing reproducible code, you can start practicing by using public data. So you don't need to wait till you have funding to generate your own data, but you can actually go online into any of the repositories for, depositing data, and then you can just pull down any of these public data sets and, start working with them and even answer some questions that you're interested in using public data. And some of these repositories are Sequenced Archive, Geo, ENA, RefSeq, GenBank. One thing I would like to emphasize is the standard of research. So I like to encourage open science as much as possible because it improves the accessibility, the quality, and the efficiency of science. So as we do all these projects in building out pipelines or trying to find unknown functions, as much as possible, when we make our results open, it prevents other labs from repeating something that has already been done by your lab. And then the science can move forward quicker that way. So open access articles also, even though they can be expensive, but they are recommended so that those people that don't have the funding to subscribe to the journals can still access your articles and be able to find the data and interesting results you have in your paper. So the research data, the code, and the pipelines should be fair, meaning that they're findable, accessible, interoperable, and reusable. And as much as possible in your code, document you had comments because it helps you in the future, and it helps other people that also want to understand what you are doing when you were developing the code. So data sharing is very important. If you're not sharing our data when annotating these unknown genes, it will take longer to annotate the unknown. So as much as possible, let's share data. Thank you very much.
Dr. Samantha Mirage leads the genome editing program at the National Institute of Standards and Technology. She was awarded the Outstanding Young Scientist Award for Non-Academics in 2019 and received her doctorate from the Johns Hopkins University School of Medicine. Her presentation is entitled Quality Control and Standards Overview.
Hello, I'm Samantha Mirage from the National Institute of Standards and Technology, and I'll tell you a little bit about how you can begin to think about QC and standards for your processes. So, you all are early on here at development, and what you want to be able to do is not fall into these translation valleys of death. So I'm going to try to convince you that standards and QC is going to help you shore up to make sure that you don't fall into a place where you can't translate the work that you're doing. Reproducibility. So this is a big word. We want to reproduce our data. We all talk about this. What I want to sort of break a little bit here is I want you to think about how confident are you in your data? Should you trust your data? Can you trust it to make important decisions? So reproducibility may give you precision, but it may not be accurate. There can still be biases. There can still be things here that are giving you the wrong impression. And so I want you to think about, what do you need to have high confidence? Reproducibility is one factor of that, but I want to break this idea open and say that there's going to be variables pre-analytical, so things in process, shipping, things like that. Analytical, maybe there's noise in the assay, and that might give you some challenges or present itself like its biological signal. And what you want to get to is, do I believe that I can trust this data to be representative of my biological system? So here are some key things I want you to think about. How confident are you that the answer is correct? What's the consequence if it's wrong? And what can be done to understand if the answer is correct? And I don't want you to just apply this to the work of this program. I want you to apply this broadly as you do science. Okay, so standards. Standards, I want you to feel like this is going to help you. Standards are not disabling. They're not going to hold you back. They're not going to limit you. Standards do just the opposite of that. They're going to empower you. They're going to help you accelerate and learn faster. They're going to you're going to help you know whether you're getting confident data, and they're going to help with comparability, translating to others, or as a new technology or a new update of your technology comes forward. So standards can be any sort of thing that allows you to have some sort of concrete hold, something that's shareable content-wise, that can be used to make sure that you understand whether your process is working. They could be practices, terms and definitions, materials, physical things, requirements, and things that aren't even listed on this slide. So you're all at the beginning here of these assays. And on top of that, I want you to begin in your mind to layer the idea of making sure you've got good experimental design. What control strategies, what kind of testing strategies could you do to make sure that your assay is actually reliable? So I'm from NIST. We're the National Institute of Standards and Technology, so I want to tell you a few things about us. We're the measurement people. You might not know that we're here, but we're here supporting from the U.S. federal government side. We're non-regulatory. We are not functioning primarily as a funding organization. We are the measurement scientists that are there in the trenches with you, in the labs, doing the measurements to try to help you understand what controls, what would be useful, what we can develop to make sure that key kinds of assays that the U.S. economy really needs, that people can know that they're giving reliable information. So we work, it's just a quick example, we work all across different sorts of organizations, helping to try to fill and not fall into those translational valleys of death. And we work across all different sorts of assays. Here's just a snippet of some of the ones in the biological sector that are within my unit. So one of the ways that you can have better confidence is control materials. I just want to pop up here some kinds of control materials that we have available that you may want to reach out if you're interested in. And if you're interested in working together with NIST, we do have these 4 active consortia where we're collaborating directly with users to develop controls and control strategies. So when you're talking about your work, where can variability happen in your process? I'm going to give you the answer. The answer is just about anywhere. So as you start at the beginning of your process with sample collection, as you store, handle, you do analysis, you analyze data, all across this process you can get variables that are not related to your biological function. So you want to put into place strategies that help you understand whether you're getting variability that doesn't really have to do with the test, the experiment, the thing that you're trying to detect. And in the end, you're going to make decisions, and you want to know whether you trust that data to make important decisions. I'm just going to flash up here some examples of some strategies that might help you at these different parts in the process. So I mentioned this word before, measurement assurance. It was on that slide, that triangle graph. Measurement assurance just means how confident are you that you can trust your data, and the strategies that you might put in place to make sure that you're confident. So how well do you have to measure? So measurement assurance, here's some tools that allow you cause and effect diagrams, what's going on, looking through design of experiments, really thinking about that carefully, orthogonal measurements, using more than one technique to try to get something, control materials, process controls in our lab studies that allow you together to have a sense of how confident are you in your data. Should you be making important decisions based on your data? So what are you measuring? Is your assay measuring what you intend? Are there variables that you need to account for? Can other people get the same result? Know your assay. Okay, so 2 quick caveats here. 1, in the work that we're doing, I would be shocked if there isn't some sort of informatic process in what you're doing and the approach that you're taking. You need to know whether that process is reliable. You need to stress test that as well. You need to record those parameters, make sure they're documented, and that process is tested. Note 2, many of the processes that you're doing may involve in silico assays or prediction. I want you to think very carefully about this. In most instances, the information that goes into those in silico approaches can't recapitulate the full complexity of a biological condition, and you may be misled a little bit. So I would propose that you really think about whether you can take the information from those in silico approaches in place of biological verification, and I would say that verification will give you even higher confidence and may even be necessary to know whether something like function and annotation is really what you think it is. In biology, it's so hard to find ground truth, but you need to make sure that you're comparing things to the right comparator in your process. That if you're annotating something in E. coli, maybe using a human genome control might not be the right thing. So it's got to be relevant to your system that you're analyzing. So since ground truth is hard to find in biology, you've got to use strategies. You might need to qualify your process. That's understanding your dynamic range. What can your process tell you? Assay validation says, okay, I need my process to tell me 5. Is it able to tell me 5 reproducibly? And then control strategies that help you. So is your data good quality? These are things I want you to think about. These are separate concepts. Precision is what we might call reproducibility. Accuracy needs some ground truth. Robustness, what's the range in which this assay might actually be run, or your cells might actually live and function to know whether you're getting assays that could report out within the parameters that are realistic. Limits of detection, having controls, and reporting. It's really important to make sure that you document. This is one of the places where we find the biggest gaps. There wasn't enough information. We couldn't reproduce it. So this is just a very quick example from NIST showing cholesterol measurements. And what I want to highlight here is the use of control materials, and that the assays and the dispersion between different labs got smaller and smaller as the reference materials were more and more relevant to the use case. So powdered cholesterol versus cholesterol in serum versus cholesterol in human serum versus cholesterol in those matrices at decision points that are medicinal. So really, the more relevant, the closest to your actual use case system you can get your control materials and your reference materials, the more reliable they're going to be to give you information that is usable. So experimental design is critical. So I want to drill this home. No volume of data, number of replicates, and statistics can tell you information that the assay design was not meant to be able to tell you. So here's a little cartoon. You can read it. But if you are doing the wrong thing or asking the assay to do something it can't tell you about by the way that you run it, then you're never going to get reliable information. And you just need to really think very carefully about this. So identifying sources of uncertainty, I just want to show you this in an Ishikawa diagram or a cause and effect diagram, where you can diagram out your process and say, where are places where I might need to pay attention? Here's an example of one. My work, I lead the genome editing program at NIST, and so this is a recent paper we put out where we diagrammed out if you were not getting genome editing, here's some ways in which maybe, you might want to think about where you could have had process failures. So that sort of concept with your assays. Reporting minimum information, reporting standards, that sort of thing. Here's another example from out of our lab. And here's where I want to leave you with. I want to leave you with points of discussion Think about this. I'll leave this up here. What types of assays are you doing in the future? This is all inspiring for standards and controls. Wouldn't it be great if all of these kinds of questions you had answered because you put in controls, you put in place processes that allowed you to know all of these things reliably about your process, your new technology, and everybody could use it, and we could get wonderful things and knowledge because of the annotation and function that we're going to get from the kinds of things that you all are doing. And we're going to be able to use that, and we're going to be able to learn because these kinds of concepts you thought about early and put into place strategies. So thank you, and please reach out if you have any questions, and I'll leave that to Renee to facilitate how she would like that done.
Dr. Kristen Burnham-Johnson is the group leader of Functional and Systems Biology at the Environmental Molecular Science Laboratory located on the Pacific Northwest National Laboratory campus. Her research is dedicated to achieving transformative molecular-level insights into environmental and biomedical systems by implementing advanced mass spectrometry instrumentation. Her presentation is entitled Beyond the Genome, Multiomics Across Scales.
Hi, everyone. My name is Kristen Burnham-Johnson. I am a group leader of Functional and Systems Biology at the Environmental Molecular Sciences Laboratory. We're located on the Pacific Northwest National Laboratory, or PNNL campus, which is located in Washington State. And what I'm going to talk to you about today is multiomics across scales, so really bridging that genotype to phenotype knowledge gap. Biology at the most basic level is the study of how living organisms function in their environment. To understand genes of unknown function, we want to characterize the phenome. So you're probably asking, well, what is the phenome? Well, as you can see stated at the top of the slide, it is the set of all phenotypes expressed by a cell, by a tissue, by an organism, or by a species. Now, conversely, the genome, or all genes in an organism, it acts as an instruction manual where different instructions can be given for each environment. And these instructions are what guide unique phenotypes. In this simple example on this slide, you can see how we have corn genes plus environmental conditions lead to a phenotype, which in this case is corn kernel color. So due to the dynamic information, our ability to read DNA, it really far surpasses our ability to understand the information it contains. The vast majority of genes have unknown or non-validated functional annotation for proteins encoded. As an example on this slide, pictured is E. coli. The E. coli genome was completely sequenced back in 1997, so 27 years ago. Yet only approximately 58% of the genes or the genome contains validated function. So that's what we know. The remaining approximately 42% of the genome remains functionally unannotated. And how is the genome dynamic? How does it provide different instruction manuals depending on the environmental conditions? Well, it does this with downstream information. The biological flow of information goes from the genome to the transcriptome to the proteome to the metabolome. And at Pacific Northwest National Laboratory, our research focus is on the downstream information, so proteins, metabolites, lipids, and how they directly impact phenotypes. Now, these, of course, are not easy measurements, and that's what we're trying to show with this slide. So the purpose of this slide is to emphasize that the proteome is complex. We're getting an increase in information as we move from genes to messenger RNA to proteins to proteoforms, and these proteoforms contain post-translational modifications depicted in the purple box in the middle. Now, these directly impact the activity of the proteins and how they impact phenotype. But characterizing proteins and their post-translational modifications is not enough to understand the phenome. So remember, genome plus environment equals molecular networks. So it's these molecular networks which in turn determines phenotype. So we go beyond a parts list to understand interactions, regulatory, enzymatic, and signaling interactions, where metabolites are often the intermediates and the end products of these biological networks. What tool are we using to measure and understand biological networks? Well, we're using mass spectrometry. I'm not going to go over the workflows, but the purpose of this slide is to illustrate that the workflows have multiple steps, so we can benefit from future high throughput methods and developments. And of course, if we want to capture multidimensional biology, which exists for all complex systems, one static measurement is not enough. We want to map molecular networks across time, across space, across different environmental perturbations, and then only then can we characterize genes of unknown function and understand that phenome. And why study molecular networks in a spatially constrained manner? Well, most phenotypes, they're observed at a global level. So many cell types or species, they contribute differently to the global phenotype. And if we increase that spatial granularity of the measurements, that enables us to understand how each component of the system contributes to the overall phenotype we want to understand. As an example, I'm going to highlight the study pictured at the bottom of this slide. So this is a microbial community that is very efficient at breaking down plant material, but it's heterogeneous. So in order to understand the overall process at the molecular level, we had to zoom into microscale activity zones. So this microbial community is the leafcutter ant fungal garden ecosystem. So how exactly are we analyzing microscale activity regions of plant degradation in the system? We're using a new approach we developed, which is called the metabolome-informed proteome imaging approach. We're analyzing 12 micron thick fungal garden sections with lipidomics, metabolomics, and proteomics, so we can map what microbes, what enzymes, proteins, lipids, and metabolites, and what activities can be correlated with micron scale regions. And this provides us with mechanistic knowledge on how lignocellulose is degraded in this ecosystem. And further details can be found at the publication at the bottom of the slide. So first step, for spatial metabolomics, MALDI imaging allows us to profile metabolites with a spatial resolution of 50 microns and correlate morphologically unique features with metabolome hotspots of interest, which in this case is the lignocellulose degradation. So we perform spatial metabolomics with MALDI, and we identify our hotspots or activity regions of interest. Then we go on to step 2. We perform spatial proteomics on these same regions. We use laser capture microdissection to liberate and move each tissue region into our micropods chip, where proteins are prepared for mass spectrometry analysis. Using this novel metabolome-informed proteome imaging approach, the spatially resolved data is able to visualize pathway-level information on the microscale by detecting metabolic activities and identifying microbial community members and their precise enzymes responsible for these activities. For our microscale multi-omic mapping experiments, for metabolomics, we mapped hundreds of unique metabolite images, which really revealed that heterogeneous spatial distribution of various molecular features across activity zones of the fungal garden sections. Then for proteomics, we detected over 7,400 unique and taxon-specific peptides in these activity zones. And we're particularly interested in lignin degradation activity. So as you can see on the slide, lignin is a complex organic polymer. It's made up of aromatic compounds in plant cell walls. And we found that our microscale lignin degradation activity zones were localized to the blue, yellow, and red microscale regions highlighted on the left of this slide. In using that information, we were able to depict multi-omic pathway level data on the microscale. So here we're highlighting one of our pathways associated with lignin degradation, specifically aromatic compound ring cleavage, where the images are shown for each of our metabolites, So the substrate on the left, the product on the right, and if you direct your attention to the middle, that's our fungal enzyme responsible for this precise ring cleavage. And as you can see, this fungal enzyme, it co-localizes to our activity regions. So the blue, the yellow, and the red regions, they had high spatial correlation with the product. What's my future dream for this research? Well, really is to move biological understanding from phenotype all the way to the phenome. So to do this, we want to make dynamic measurements of altered systems and see how this affects molecular networks. We want to keep doing that, and that's that middle circle in there. We want to, in an iterative fashion, keep doing this until we have knowledge. We have that knowledge base to predict how changes will affect molecular networks. But as you saw on one of those previous slides, multi-omic workflows, they have many steps and many targets. So current setups are well suited for the blue, the level 1 and 2 phenotyping, which are focused on a smaller number of targets where low functional knowledge is unlocked. But what do we want? Well, we want level 3 and especially level 4 phenotyping, where high functional knowledge is unlocked, so in the green. But to do this, a very large number of targets, a very large throughput is needed. So our current multi-omic workflows need to be improved. We need automation. We need fast omics with high depth of coverage. We need systematic dynamic measurements. We need advanced computational approaches such as machine learning and artificial intelligence to really deal with the big data and make sense of it. And with that, I'd like to thank you all for attending my presentation.
Dr. Adam Deutschbauer is the department head of the Comparative and Functional Genomics Group in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley National Laboratory. He develops next-generational tools for microbial functional genomics. His presentation is entitled Characterizing Bacterial Genes with Large-Scale Genetics.
Hi, I'm Adam Deutschbauer. I'm at Lawrence Berkeley National Laboratory in UC Berkeley, and I'll talk about our work on characterizing bacterial genes using genetics. So it's not news to anybody. There's many sequenced bacterial genes that we don't know what they do. This is from about 10 years ago, but things haven't changed much. There's only a small fraction of bacterial genes that have been sequenced that have been experimentally characterized. Our approach that we're applying, I have quotes, is high throughput microbiology. I'm a geneticist. I mostly use genetic approaches to infer the functions of genes from their mutant phenotypes. As part of our work, we study many different bacteria. And so because of that, we constantly think about how we can miniaturize our assays and drive down costs. All the things I present afterwards are not just my work, but it's part of a collaborative effort with multiple teams at different institutions. I'm fortunate to be part of 4 big team projects. The top 3 are all funded by the U.S. Department of Energy, and the bottom one is an NIH project. So I'll first talk about an approach called RBTN-seq that we use extensively for characterizing gene function in bacteria. I'll go through 6 challenges that we face in our own work and how we're trying to overcome those challenges. And finally, a single slide on my opinion about how I would fund an effort in gene function discovery in microbes. So we make extensive use of transpose on-site sequencing. I didn't invent this approach. The authors of this review are the pioneers. And this is a great technique. The idea is that you can use a transposon to make an insertion mutant in your organism. You can actually make a large population of these mutants such that every gene is mutated. And then a key insight is that you can pull all of the mutants together and you can measure the importance of different genes to different phenotypes by competing the mutants together in a pooled fitness assay. And you can measure the mutants before and after selection using next-gen sequencing. The the adjustment to this that my group made was to put random DNA barcodes, just random 20-mer sequences, inside the transposon, and we call this RBTN-seq. And so when we measured the importance of all of the genes and the mutants in a mixed population, we just sequenced the DNA barcodes before and after selection. And so we call this barcode sequencing, BARSeq, and it's extremely scalable because we can add all of the Illumina adapters directly to the primers. So this, in essence, eliminates the Illumina library prep cost. And so we use the exact same common priming sites for barcode sequencing for lots of different methods that ourselves and our collaborators have developed. And these different techniques are down here in blue, and I'll just talk about mostly RBTN-seq today. So this is a few years old now, but we were able to demonstrate that you can take a data-driven approach to better characterize gene function in bacteria. And so in this study, we did around 5,000 of these RBTN-seq assays across 32 bacteria. And we were able to identify phenotypes for many, many genes that we had no previously known function for. And in over hundreds of cases for enzymes and transporters, we can make a very specific annotation based on the pattern of its phenotypes. So what are we doing now? We're both expanding the number of strains we're investigating with this approach, and we're expanding the number of conditions that we profile as well. And this expansion is possible because the cost of sequencing just keeps going down. And so on the latest and greatest Illumina machine, we can sequence over 3,000 of these assays at a cost of under $4 per sample. So challenge one that we face is getting genetics going in different organisms. It's probably not news to most microbiologists, but getting genetics to work in gram-positives is a little more challenging than gram-negatives. There's different solutions to this. One is to just try many different DNA constructs against an organism at one time. And so we published an approach like this a few years ago that we call magic pools. And you can basically barcode vectors and have different promoters and drug markers, and you can figure out what's the optimal construct for doing genetics in a given organism by just sequencing the barcodes. Other approaches include overcoming host defense systems or improving DNA delivery methods like electroporation. The second challenge that we have, and this is sort of like a community-wide challenge, is how do we propagate the knowledge that we gain from doing our assays and new gene annotations to new genomes. And so like traditional annotation and biocuration by established databases like UniProt sometimes isn't straightforward. Another is just propagating our knowledge to new genomes and to new annotation pipelines. One solution that we're taking for certain pathways is called Gapmind. So we have a tool for this for both amino acid biosynthesis and for catabolism of smaller carbon sources. And here we take experimentally characterized proteins for each step of a pathway, in this case, the pathway for making serine, and we supplement the known proteins that do that activity with the knowledge that we gain from doing all of our genetics assays. And so we can then quickly go through any new genome and ask, are there reasonable candidates for all steps of this pathway that support the presence of that pathway? The third challenge that we have is that the pooled assays that we do aren't ideal for every phenotype that you might be interested in. For example, genes involved in secreted factors are not ideal in these pooled fitness assays because you can get cross-complementation of the mutants. We've done many of these assays. They're not particularly informative for understanding secondary metabolites, for example. So one possible solution to this is actually not doing the assays as pooled mixtures, but to assay individual mutants where there's no crosstalk between the different strains. And my collaborators at Stanford, Anthony Shiver and Casey Wong, are working on these kind of approaches. And so in this study, they made an ordered collection of individual mutants for nearly every gene of Bifidobacterium. And were able to use microscopy to look at cell shape of all of the mutants, as well as metabolomics. The fourth challenge, which I'll touch on really quickly, is that we do a lot of chemical genomics. And very often the compounds that we want to use or assay, and very often intermediates and pathways are compounds that are just not commercially available, or they are available and they just cost a ton of money. So one solution is either spending a lot of money or maybe partner with chemists who can make some of these compounds. It's probably not news to most people, but most bacteria that exist in nature are currently uncultivated, and so we're missing a significant fraction of the gene space using our standard kinds of assays in a laboratory. So one solution to this is just doing genetics directly in a community context. And my colleagues at the IGI Institute at UC Berkeley are pioneering these kind of approaches using CRISPR-guided transposons. Another approach is just expressing heterologous DNA in a host and doing assays. And so Yolanda Wong and Adam Arkin have developed an approach like this, where you can express DNA from different organisms in a host like Bacteroides theta iota-micron. The last challenge is how we make our inferences about gene function from mutant phenotypes. Nearly everything we've done to date is just by individuals staring at the data to make the inferences. And this isn't scalable, especially at the rate that we can generate data now. And so probably the most obvious solution to this is some kind of AI machine learning. And it's exciting that there's approaches like this coming online. These are two different groups that did machine learning kind of approaches to predict enzyme function, specifically predicting EC numbers. It's a little different like how they did the math behind this, but both of them had similarities. They both use SwissProt for their training sets. And both of these studies also use us to test their performance. They tested their performance on bacterial enzymes that we had annotated using our genetics data. And so how did they do? They work pretty good, but there's room for making it better. And so when you try to predict a more detailed EC number, the rate of correct predictions drops. And in the other study, it worked pretty well, but it wasn't substantially better than using BLAST-P. All right, for my last slide, if I were funding an effort to characterize bacterial genes at scale, I'd fund a network of people to work on this. And for those who have like core technologies that are mature and ready to be applied at scale, say TNC, metabolomics, proteomics, I would apply them at scale across thousands of diverse bacteria. And it probably would make sense to engage the community for their favorite microbes and experimental conditions, and to provide everything, the data, the genetic resources, all free of cost and prior to publication. I think there's room for lots of new technologies that can be applied across thousands of bacteria at scale and at low cost. And so I would fund high risk, high reward tech development with the charge that's written here. And there's probably lots of things that could be done. So protein-protein interactions, gene regulation, double mutants, genetic epistasis, structure function. And if anything really panned out and was scalable across a very, very large number of genes and proteins, then it could be blended into this core program. And then the final thing, which I have spent many arguments with people over, is I wouldn't spend much time trying to mine existing data from the literature and trying to pull old gene expression data from Excel files from some paper from 20 years ago. I think that's just fraught with problems. I just generate new data at scale, and there's sort of an advantage of generating data within core teams that have agreed upon for metadata, how you describe your genomes and organisms and your compounds, that would ensure that all of this is cross-comparable and machine readable. Thanks.
Dr. Zachary Sholop-Powers is currently the Research and Development Director at Ginkgo Bioworks. His past research focused on the interface of metagenomics and drug discovery. His presentation is entitled Identification and Prioritization of Biosynthetic Gene Clusters for Commercial Metagenome Mining.
And I'm going to talk, as one of the few industry participants, I'm going to talk a little bit about the company I work for, Ginkgo Bioworks, some of their capabilities and how we think it can be brought to bear on the problem. I'm going to give a little bit of my history in using, basically mining the unknown metagenome for the purpose of natural product discovery. And then I'll close with this slide that Renee asked for, which is about what would you like to do going forward to solve problems in this domain. And here we go. So the background I'm going to give a little bit is around Ginkgo, which is a full stack synthetic biology company. We've got capabilities from computational, fermentation, strain optimization, and scale-ups. The headquarters is in Boston, but we have a presence on the West Coast and in Europe as well. And each of these teams have different specialties: a fungal team, the FGen team, ag bios on the West Coast. We have a number of, I think, unique or distinguishing capabilities that we'd like to deploy to projects and potentially a project with you. One of these is a relatively large and unique metagenomic library that's built up over years of sequencing from companies that Ginkgo has acquired, as well as from internal sequencing efforts. And you can see here, we've got on the ballpark of a billion genes, and that number has been growing. This is very novel, less than 5% when co-clustered with industry. So I'm sorry, with all public databases. So we've got a unique metagenomic sequence database that we can deploy to problems, and I think that's quite nice. We'll get into that a little bit more later. Additionally, that deep sequence metagenome not only has proteins, but it also has synthetic gene clusters. You can see in our current database, which primarily in this case is from Zymergen and Warp Drive, is on the order of 5.5 million gene clusters. Additionally, Ginkgo has a large screening capacity. So there's a traditional screening center. There's 10 robotic work cells. You can do thousands of assays a week. But what I think is really a distinguishing capability is the nanoliter reactor platform, which basically can allow you to encapsulate basically anything you want into single, you're not really cells or reactors, alginate beads, which then can be assayed for fluorometric properties. So this is really beautiful. It allows you to have a lot of flexibility, sequencing at the level of an organism. This is an example of showing on the left-hand side a couple of different types of organisms, E. coli, Cerevisiae, chlorella, as well as a couple of different applications one could consider looking for proteins, chemicals, biologicals, or cells, as long as they're tethered or coupled to a fluorescent readout. So Ginkgo is also heavily involved in automating as many processes as it can. And there's a wide variety of hardware available to scientists within Ginkgo to automate and scale up any of the processes they're working on. And of course, some of these are internal research projects, but also these are deployed for collaborative ventures with scientists like yourself. So one of the I think premier ways of deploying the capacities above is in the enzyme space. So this metagenomic sourcing, this metagenomic sequence data asset can be deployed for selecting or finding protein sequences from nature that have improved activity relative to baseline. And so we've been able to use this in a wide number of projects to improve enzymatic activity, and typically that's upstream of traditional protein cycle of optimization of protein. This is a pretty typical case study where after 4 rounds of engineering, we're at 10x of an already somewhat optimized protein. This is another look at biocatalysis, and we can see again a little bit closer where we're targeting those mutations, inactive site residues, or key contacts. And again, we typically see, in this case, 6x improvement. This is a nice and pretty standard improvement. And one exciting thing that has happened last year is that in addition to this sort of traditional assay-based wet lab capability, we've been growing out our computational resources. And I believe in August, we announced a pretty significant deal for access to cloud compute resources via Google. And we're deploying this for a couple of things, most notably and publicly the creation of a protein foundation model that we're hoping to deploy for all of our projects. So that's a little bit about the company. We're going to turn a little bit now to this, to the topic of the seminar, which is domains of unknown function. And we're going to talk to you a little bit about some background in a couple of natural product discovery companies that I've been a part of, Lodo Therapeutics, Zymergen. These are both companies that were acquired by Ginkgo, and Warp Drive Bio, which I'm not part of, but we're also acquired by Ginkgo. We have a lot of scientific expertise in this area. But the basic logic of metagenome mining or genome mining is that natural products are these fantastic molecules with exquisite biological activity. Because they're created by biological creatures, they are assembled via biochemistry, and that biochemistry is ultimately coded as genes in their genome. If there are unexplored genetic spaces, it means there might be genes that can encode for new molecules, and there may be environments where that genetic space is vast. That genetic space is vast, can be mined, and we can potentially find new compounds. And so the twist for these different companies that Lodo basically went around to gather up lots of different soils. Zymergen was willing to put up the cost to deep sequence all of the libraries that we created from those soils. And there was success for all those companies in terms of being able to reach milestones on deals with our pharma partners. And so whether on the left-hand side you're starting with a metagenome or a culture collection, you still have two major choices, which is to analog, to find gene clusters that are similar to the ones that you're interested in, that you're starting from, or to try to identify clusters that might have a property of interest, and typically that's determined by the presence of a gene that might act as a resistance mechanism, and therefore be a decoy, and therefore be homologous to the target of interest. So you try to pre-assess what the target of the compound is going to be based on the presence of the resistance gene. Those two main strategies, and downstream of that, the selection of the cluster, the isolation, and analysis is basically the same. That's the process. And so how did Lodo do it? So basically, this is the way that we did it at Lodo. A nice example of success. We took 2,000 soils, isolated small amounts of DNA from each one of those, used PCR to generate these tags, found a specific family of tags that we believe were responsible for one of the amino acids in this GDXD calcium binding motif. We are then able to, on the top here, I don't know if you see my mouse, be able to build an entire array DNA library, use PCR to find the specific clones, recover and assemble those clones into a full gene cluster, heterologously express, ferment, isolate and characterize the molecule. We were, in this case, to come up with these two compounds, maloside A and B, which had a unique mechanism of action. So this is a beautiful case study about the potential power, of the rare biosphere to offer up new antibiotics. At Zymergen, we basically were allowed to scale because we sequenced quite a bit more. And this beautiful spinning picture is demonstrating a million biosynthetic gene clusters, all co-projected based on their distances to a set of known gene clusters. And what we're seeing here is basically clusters colored by the class, and unsurprisingly, the classes are separate. And so this is kind of a nice picture, but it can be deployed for analoging purposes. So within Zymergen, we did focus primarily on the resistance gene type compounds, so using these decoy homologs. And this is an example using I forget what the DARPA was here. MetaP1, there you go. Using MetaP1 homolog to identify clusters of interest, to categorize, retrieve, to engineer the microbe, to find the metabolite, to characterize the molecule, and ultimately we were able to show that it's a binder of MetaP1. So this is another nice example. So this is great, and this shows the possibility of using uncultured organisms for which you do not know much about them, and use them to have some useful commercial activity. But there are some drawbacks. Obviously, we were acquired, and so there's, I think, 3 take-home messages that I'd like to highlight relative to relevant for this crowd. The first is that analoging, while feasible, is less commercially viable. It's unlikely to find a first-in-class compound, so it's hard to get financing or find a good market for compounds that come out of that class. The second is that the embedded resistance gene can work, but it's not guaranteed to work. Certainly not guaranteed to work on all the targets that are, bring in significant dollars. So the commercial interest for these two is pretty difficult. But the fundamental problem is that while the sequencing costs have dropped per base, the actual conversion of BGCs, which are cheap to obtain, into compounds, which is still very difficult. And so that's not great. And so I have some recommendations here, right? We can solve those problems, but fundamentally, if we want to do a better job of characterizing all of these unknown sequences, all these unknown metabolites, we need something down here. We need a way to drive down this cost curve of characterization. And so this is a little bit coming back to Ginkgo, which has some of these high throughput capabilities. And so the dream side would be, I think, just highlighting at a high level what we did well in natural product discovery from the metagenome is there are many undescribed VGCs in the metagenome. Within Ginkgo, we have this fantastic screening facility. We have a great enzymology team, and I described to you this major investment in AI dataset creation, dataset generation, model creation. So we got these great things, but we still have this main problem, right? Large-scale characterization in VGCs via traditional culturing. And so basically the characteristic of the type of project that I would imagine would be a good match between the desire of this team to characterize unknown, the functional manifestation of this unknown biochemistry with something on the industrial scale of Ginkgo would be to come up with a series of projects that do a deep characterization of aspects of biochemistry that are really, really important for biosynthesis and discovery. And so what I'm just proposing here is two. This is not a full end-to-end pipeline, but this is two aspects of enzymes that I think someone like Ginkgo could potentially deep characterize, which would be biome-wide adenylation substrate characterization and biome-wide thioester substrate characterization. So for non-ribosomal peptides, basically the sequence determination of what will get incorporated within a non-ribosomal peptide is going to be determined by the interactions between the adenylation and substrate. We can actually go out and take all the adenylation domains that we've ever characterized, cluster them, and characterize them across some number of some reasonably large number of substrates, and then come up with sort of the best in class predictive capability for adenylation. I think that's something that would be very useful for predictive capability. And second of all, I think cyclization is still an open question. I think we have a lot of potential cyclization proteins and domains that we could choose from, and the high throughput assay. So I think that this, if we can take the difficult problem and make it soluble and approachable to our strengths, I think we have a really great way to go forward. And so I'd like to thank the organizer, and I'd like to thank everyone here for listening to the talk. It's been a really great day so far, and look forward to chatting more. Thank you.
Dr. Andrila Mukhopadhyay is the Division Deputy of the Biological Systems and Engineering Division at Lawrence Berkeley National Laboratory. Her research involves microbial engineering for the production of biofuels and bioproducts. Her presentation is entitled Genomics-Aided Host and Strain Engineering for Biotechnology.
Hello everyone, I'm Andrila Mukhopadhyay, a senior scientist at Berkeley Lab, and I'll be talking today about our genomics-aided host and strain engineering work for bioproduction. So most of the work that I'll present today was done as part of large multi-team projects at Berkeley Lab, and it spans the work done for microbial communities to those in engineered microbial systems. Specifically for JBay, our goal is to convert complex carbon sources via deconstruction using microbial platforms to biofuels and bioproducts. So lignocellulose is one of many such renewable carbon sources that can be converted to desirable targets. For this, we have access to many microbial platforms with various capabilities and catabolic profiles. In our case, we are converting lignocellulosic hydrolysates to hydrocarbons that serve as biofuels and as bioproducts. And one such example that I'll be telling you about today is indigoidine. Indigoidine is a non-ribosomal peptide. It's a secondary metabolite, which is a dimer of two glutamines. It is a bright blue pigment that can serve as an alternative to indigo, the chemical manufacturing of which comes at a pretty heavy cost to the environment and has a very high carbon footprint. So it's desirable to come up with alternatives and for biological routes to these kinds of compounds. So indigoidine itself is a relatively simple non-ribosomal peptide, and it is relatively straightforward to engineer an organism like Pseudomonas putida to convert sugars or aromatics from lignocellulose to this compound. However, the real challenge is to scale the system up to operate in a bioreactor. And this is because a bioreactor environment can be drastically different from that of a shake flask in which we optimize our engineered strains, and the hydrolysate that we use can be very complex, dense, and heterogeneous in a bioreactor as well. So these are very unintuitive phenotypes that we are trying to engineer, where we need to have not just an engineered pathway, but also a capability to convert all the carbon into the final product, be able to tolerate both intermediates and the final products that are being produced by the system, have high flux to the desired metabolites, and maintain fitness and metabolism in a bioreactor. So for this kind of engineering, we find that functional genomics is usually very effective in providing us with the gene targets and strain designs that we need to proceed with. We use a range of approaches such as multi-omics approaches, adaptive lab evolution, metagenomics, the RBT and seq, the mutant libraries that Adam Deutschbauer talked about in this workshop. And we also take data-driven approaches when we do have good quality, systematically obtained data. And the noteworthy aspect of using functional genomics is that it not only reveals known enzymes with known functions, we often find new functions for known proteins, and we find the role of unknown proteins in these processes, and we go beyond metabolism to understand the role of regulation and signaling. So in one example where we used a transposon library, what we did is that we took an engineered strain that had been engineered for the production of indigoidine from aromatics, and we put a mutant library of Pseudomonas putida through the RBT and seq workflow in a bioreactor. This resulted in the discovery of genes that, when deleted, both give fitness and reduce fitness in a bioreactor. And the most sort of prominent target that fell out of it was not a metabolic enzyme. It was a signaling protein, a regulatory protein called GAC-A, which controls a variety of different cellular phenotypes, including attachment, etc. And when we delete this gene, the strain actually performed better in producing indigoidine, but it operated or it performed significantly better, substantially better when we examine indigoidine production in a bioreactor, where we got really good production. So in a single study using functional genomics, we were able to go all the way from microtiterolates to shake flasks to a bioreactor for the production of a bioproduct from an aromatic intermediate. So another approach that we use very efficiently are genome scale model-based approaches. So we use genome scale models to come up with strain engineering design comprising of gene edits, which when implemented not only improve the flux of the carbon from the substrate to the final desired product, but also couple growth or substrate utilization to the production of the key intermediate. intermediate. This gives us very robust production parameters that are maintained across scales all the way to bioreactors. And we've successfully implemented these kinds of approaches for sort of well understood carbon sources like glucose. The place where we hit a challenge is when we are trying to use these approaches for less understood catabolic targets like aromatic compounds from lignocellulose. Here we found that implementing the design that we got from the genome scale model resulted in a strain that was very inhibited. So what we did is that we used adaptive lab evolution to improve the growth of the strain, and then used proteomics to understand the context of this gene design implementation. And using the proteomics data, we could build context-specific genome scale models that then resulted in designs that were much more successful. And the final strains from these studies were useful in not only producing a high level of the final product, but could produce this product from hydrolysate, where the basal strain couldn't even grow in the hydrolysate. And finally, I wanted to tell you about some resources that we found for engineering new organisms by studying environmental systems from the Oak Ridge Field Research Center. This is probably true of many environments and microbial systems that we can study. But in this particular project, we were examining this environment for mobile genetic elements. And we ended up isolating a large number of plasmids from groundwater samples of this site. These plasmids not only spanned a very large range in size, but also were common across many different wells in these environments, and ranged across all the different incompatibility groups that we work with. So what we did is that we took the most ubiquitous plasmid that we found across many different samples and screened them against isolates from this environment. And we found that this plasmid could be naturally transformed into these organisms. So what we have is basically the first tool to transform novel isolates. What we also did is that we took this set of plasmids and screened them for origins of replication. And where we could predict an origin of replication, we built a library of these parts in plasmid scaffolds in a barcoded manner, and again screened them against new organisms. And in this case, we could find new origins of replication that could be used to transform novel isolates, not only to express same model proteins like GFP, but actually express heterologous pathway for biomanufacturing. All this is to say is that functional genomics can be a really powerful tool to discover gene function for both known and unknown protein in the context of engineering microbial systems. And we can develop microbial chassis for a variety of applications, including degradation of harmful substances, biomanufacturing, biosensor development, and discovering targets that are useful for applications in therapeutics, precision communities, ag applications, probiotics, etc. So with that, I would like to thank my group that did all the work that I presented, and our funding agencies that support the research that we did. Thank you so much for having me in this workshop.
Dr. Iran Nagman is an assistant professor in the Department of Molecular Biology and Biophysics at the University of Connecticut Health. His lab focuses on developing comprehensive multiscale models of cells. His presentation is entitled Integrative Multiscale Modeling of Cellular Systems.
My name is Iran Agman, a professor in UConn Health, and today I'm going to be telling you about integrative multiscale modeling of cellular systems, also referred to as whole cell models. I'm going to start with a quote from a science hero of mine, Harold Morowitz. In 1984, he wrote, "A computer model is feasible, and every experiment that can be carried out in the laboratory can also be carried out on the computer. The extent to which these match measures the completeness of the paradigm of molecular biology." That is to say, if we capture our knowledge of molecular biology in a computer model, simulate it, look at the results and compare those to the experimental observations. The extent to which those match is demonstrates that we've understood something about the cell and the molecular biology. The extent to which the simulation results diverge from experimental observations is a measure of the incompleteness of molecular biology, and it indicates there's some work left to do. So a short history of whole cell modeling. This goes back at least a few decades, to 1973 with Francis Crick and Sidney Brenner writing about the complete solution to E. coli. Close to a decade later, Harold Morowitz advocated for a model of mycoplasma. At the same time, some efforts began, first with ODEs, some more gene-based models, and in the 2000s, coming upon genome scale metabolic models focusing on the metabolism of E. coli and other microorganisms. In 2012, the first reported whole cell model was published of mycoplasma. And in the last decade, there have been many more models come out of different organisms. In 2020, a large-scale mechanistic model of E. coli was published. This is a project I was a part of, and you'll see a little bit of that today. Okay, so this is the whole cell model of E. coli. This model combines many measurements that were recorded over decades and across different laboratories regarding all of these different kinds of data. A cell is composed of many different molecules, and there are many different molecular processes that are active, resulting in different data that's relevant. This often comes in different data formats and different experimental protocols to measure them. We connect all of this data through mechanistic models. So these are models over here, bottom left. This is showing some of them that represent the actual processes that are active in a cell. And the variables and parameters correspond exactly to measurable quantities that can be taken from the lab. And here you can see color-coded how the different data types correspond and are brought into the equations. This resulted in over 10,000 equations of the change in metabolites, RNA, protein, replication growth, amongst many others. And when these are brought together into what we call an integrative simulation, it results in a unified computational model. That computational model is simulated, we get the results, and we can then compare it to data. So critically here I show highlighted, while all the genes expressed in this were expressed in this model, so we actually create the resulting proteins and can count them, only about 43% of those genes were given a function. Most of these were metabolic enzymes and were given metabolic function, and that's critically because the E. coli model is built with its foundation in a metabolic model. And we'll see a little bit more about that. Okay, so a question came up while we were building this model. There were challenges in pulling all of this together and making sure that the model could continue to scale and grow as more data becomes available and as theory is developed for new cellular processes. And we asked ourselves, can we leverage modular software design to integrate these different data types and models of cellular and molecular function into the whole cell models? So we came up with a software called Vivarium, which was inspired by the internet protocol. We're calling it an interface protocol for connecting heterogeneous models, algorithms, and data into a hierarchical network that represents distributed interacting processes. It's ideal if the representation of the data reflects the structure of the system, in this case a cell, in which it's a hierarchical system with processes often within other processes. So here what we have in a rectangle is a process. This is the driver of the drivers of change in a system. In computationally, these are the algorithms. They consist of parameters and an update function that reads the current cellular state and provides an update. And we also have ports. Ports allow the processes to connect to different states across the system. Here in a circle is a store. This holds the state variables, and it can support any different data type, which is important because of the range of data types that are available and provided for different cellular systems. We need to make sure that this is a flexible data structure that can accommodate different data types. And so stores help us do that. A composite is a bundle of processes connected across a hierarchy of such stores, which can then be executed and run by the Vivarium engine, running them at their own, each process its own preferred time step, concurrently to support massive distributed simulation. Here we see a snapshot of the Vivarium structured graph for E. coli on the left. This is very hard to see because it's a massive simulation. So we have a little zoomed up region over here in the middle, showing a few of the processes that are present in the E. coli model, including metabolism right here in the middle. You can see its ports, how they connect to the state here. Bulk state is a certain representation of molecules. That's just individual molecule counts can be read through there. So metabolism connects to substrates, enzymes, catalysts, metabolites, reads amino acids, and so forth. And for every one of its time steps, it calculates a change that drives the system forward. On the right, you see a summary plot that takes all of the different molecules in their categories and shows how their mass evolves over time, resulting in a doubling rate of approximately 43 minutes, which matches our experimental understanding of E. coli. Because we use this modular software Vivarium with a hierarchical representation, we were able to very easily take that entire E. coli model and plug it into a simulation of kind of colony scale dynamics or higher level processes, in which each E. coli model is here shown as a rectangle. We're hiding all of the internal details within E. coli, and here it's just a process that takes nutrients from its environment in these fields, and it drives its growth. And these can grow and divide in the environment. Here you see on the right how we start off with a single cell. This is a whole cell model running inside of there, so a massive simulation in its own right. But when it grows and divides, we have many of these running in parallel. And we can track their individual dry masses. We can track the individual RNA, the different protein, and many different events. Here in E, you can see the heterogeneity resulting from this simulation, in which we have these different proteins, OMP-F, TOL-C, AMP-C, MAR in their different expression across the colony. You can see on the left, these two OMP-F and TOL-C, almost all cells express some of this, but on the right, we see some of these less expressed proteins in which we have some cells with zero, no proteins. These are what we call subgenerationally expressed genes. Okay. And now for some movies. Here is the E. coli colony growing on glucose. And it's eating the glucose. You can see glucose being depleted in the background. This is the same simulation, but we're selectively tagging AMP-C. And I'll also show AcRA-B TOL-C at the same time. So we can track any of the variables across a population. You can see here AMP-C is one of these subgenerationally expressed genes. Okay. Next thing that comes up, we can count all the proteins, and that's what we show when we tag them. But leaves us in a little bit of a bind, especially shown here with tagging of flagella. Now, here we see how flagella is expressed across the network, so all of these cells have a flagella, and so what's the catch here? You might ask, why are they not swimming? And the reason that they're not swimming is we're just counting the flagella rather than adding their function. So we need a separate model that can take the counts of a molecule and actually add function, which allows those molecules to operate and contribute to the dynamics of the cell. This is one of the main challenges we face is determining what the functions are of individual proteins and how to express them mathematically. And if there's missing function models, that's going to be missing from the resulting simulation. So here we've added function to the flagella expression. We add a process for chemoreceptors, in this case an MWC statistical mechanics model, and we have a model of flagella activity. These can be plugged in to the model using vivarium, which is really helped with this ability to plug and play, that we can experiment with different functions more easily. In this this plot to the right, this is a spatial plot of E. coli that were placed all on this green dot, and we have two different types of models. We have one that just adds the flagella activity, the motor. Those are shown in orange. And we have a different class of models that has the motor and the receptors. And the receptors are linked to the motors through a signal cascade pathway, so that changes in the environment detected by the receptor can propagate and control motor activity. You can see the resulting behavior. The motor only do a random walk, resulting in a pretty even spread from their starting location, whereas the models with the receptor and the model and the motor actually swim up a gradient, and you can see that they're much by the end of the simulation, which is these red dots, they end up higher at a higher ligand concentration. They move up the gradient. Okay, some more examples of adding functions. This is a recently published paper in which we took the E. coli whole cell model and added new processes for antibiotic transport. for antibiotic hydrolysis, for cell wall maintenance, for lysis, and a few others that aren't shown here. And we see some diagrams illustrating what these different processes do in response to two different antibiotics, tetracycline and ampicillin. Here's a video showing response to antibiotics. As the cell grow, you can see that they're growing freely, and you're about to see antibiotics introduced. There, the black is antibiotics, and immediately you see cells here grayed out that have lost the ability to grow. Their ribosomes are bound in this case, but you also see that there are some cells that continue to grow despite the presence of antibiotics, and that's because they have different protein abundance, and they happen to have the right amount of proteins that let them respond and continue to grow despite the antibiotics. Okay. What needs to happen next? In order to represent all the molecular processes in a cell, we need to integrate all of this heterogeneous data. Ideally, each of the following can be experimentally determined, but may require inference algorithms to fill in missing knowledge. So we need all kinds of data. Here I list several types of data: the sequence of each chromosome, RNA, and protein, location of chromosomal features, including genes, operons, promoters, terminators, and the location of each site on each RNA and protein. We don't know in advance what level of detail is required, and so we should leverage all the details that we can get. We think the structure of each molecule is going to matter. The domains and sites of the macromolecules, these can all be used computationally to determine function. Subcellular organization of cells into organelles and microdomains. If we're looking at more complex cells with spatial structure, that's information that's going to matter, and it's going to influence the function of the genes. We need to look at the participants of molecular interactions and the effects they have. We need models for these. What molecules are consumed, produced, transported. We need kinetics for these interactions. These are hard to measure in some cases, and so we'll take what we can, and the rest might have to be computationally inferred. Concentrations of species in the microdomains and in the extracellular environment as well, since that's going to have an impact on the cell's behavior. Okay, to connect the cell's molecular composition with its behavior and function. The ultimate goal of biology is in my opinion, we need curation pipelines that can expand upon processes developed in, well, if we're expanding in the E. coli model, we need to build upon them, adding new functions and testing how those functions determine different cellular dynamics. This includes modules for metabolism and beyond. Whole cell models made of these processes need to be calibrated with molecular data across heterogeneous cell populations and in different environments with different experimental perturbations. It's only by getting the responses in different conditions that we can identify the missing functions and add those annotations in. Okay, finally, a thank you to everyone who has helped make this project such a success over the years. And thank you to my funders. And please feel free to contact me with any questions. Thank you very much. Bye-bye.
Dr. Christopher Bettinger is a program manager in DARPA's Biological Technologies Office and a professor in the departments of biomedical engineering and of materials science and engineering at Carnegie Mellon University.
Hello there. My name is Chris Bettinger. I am a program manager in the Biological Technologies Office at DARPA, and it's my pleasure to give you a little brief on unknown protein function in whole cell modeling. And so really this is taking a step back and contextualizing a lot of the emerging and prospective discoveries in the Discovering Unknown Function program and bringing those insights and discoveries into a new domain, and that's whole cell modeling. And so just taking a step back even further, so really the field of whole cell modeling is focused on creating physics-based simulations of cell behavior, right? And so this is a pretty broad area with lots of contributors from lots of different domains. It's very interdisciplinary, but that community is informed and unified by its goal of trying to understand and realize, right, a comprehensive simulation of cell behavior. And so really the overall kind of holy grail of whole cell modeling, right, is a simulation that can predict phenotype, and not just phenotype as it stands, but phenotype from genotype, right? And so part and parcel to that is really understanding how all the molecular species sort of converge and interact, right, at a very granular level, right? And the idea is the insight from those interactions can lead to sort of an understanding of the emergent properties, right? And so this is an exciting area. It's been around for decades, and I think the potential impact for whole cell modeling is really lots of things, right? We can think about predicting diseases. We can think about anticipating the evolution of pathogenesis for certain strains. And then I think importantly also using it as a design tool to accelerate the engineering of chassis for various contexts, including biomanufacturing. I think those are really the promises that motivate and drive this community forward. And so when you think about basically whole cell modeling in this kind of white space chart, I think it's kind of well positioned, right? And so you can think about basically our ability to simulate biological systems on a log plot, and this is kind of just one lens, right? But one lens could be thinking about these models as basically to what extent are we capturing accurate physics or chemistry, and what is the relative length scale of the efficacy of those models, right? What kind of length scale can we capture with high fidelity, right? And so if you look at that kind of white space, you can kind of have on one handrail molecular dynamics in the upper left, and this is essentially highly physically accurate models of behavior of molecules at small length scales. And then you can have the opposite, which is basically something like agent-based models, which basically can be, can simulate multiple cells or communities of cells, albeit with a loss of sort of that physical accuracy. And so we can think about whole cell models as kind of operating kind of somewhere in the intermediate here, which is kind of, again, striking a balance between faithfully replicating and describing the physical behavior of those cells at a length scale that's kind of relevant and could have value for various stakeholders. And then, it's possible, right, if we can kind of expand on the state of the art and kind of push the field up and to the right, we can get kind of increasing complexity, perhaps with increases in computes. And importantly, we have increasing in the richness of the data sets that can inform those models. And I think this is where sort of the Duff kind of effort comes into play. So if we can kind of bolster our data sets in a reliable way, we can really kind of move up and to the right, which is kind of where we want to operate. And so just another way to think about whole cell modeling is is as a conceptual frame to justify and motivate understanding all protein interactions, right? And so essentially, right, we're whole cell models are all about this kind of workflow kind of shown at the right here. So it's like basically, can we think about defining an initial state of a cell? And so to some degree, or to whatever degree we can we understand and co-locate all of the molecules with reasonable confidence, with reasonable precision, right? Ribosomes, right? All the organelles, plasmids, DNA, proteins to some extent. Can we have some kind of initial state? Then if we can keep track of all the processes here shown in the point 2, right, then we can be able to sort of inform the simulations and let the simulation evolve in time with the understanding that we can kind of have and understand emergent properties that arise from treating all the molecular interactions with high fidelity. So I think that's really the excitement and the promise of whole cell models. And then again, this is data from a recent paper published in Cell, but basically, with the right amount of compute kind of conceptualized here in this graphic, we can understand and access those emergent properties. So this is a plot from simulation about doubling time, or the sort of the relative energetic costs of various functions, and also keeping track of mRNA copies in a cell as well. So this is exciting and potentially very useful. And then again, there's various degrees of success for this field so far. I think one of the most exciting advances and achievements is the ability to model a minimal cell, right? So this is sort of a reductionist version of a bacteria, for example, loosely speaking. But really, the real challenge for going from these minimal cells to these more potentially relevant but also complicated systems like E. coli, for example, is first of all a capability gap, right? This is tenfold increase in number of genes, and there's many kinds of interactions that aren't really faithfully captured that we need to capture to sort of get a grounded, accurate model. But importantly and relevant to sort of the DUF program is the idea that we don't have full observation in terms of what the proteins do, right? We don't know, we have some unannotated protein function that we have to kind of keep track of. So I think that's a knowledge gap that has to be addressed before this program, this effort in whole cell modeling can be sort of realized, or at least the outcome can be realized. And so when you think about basically whole cell modeling, what are the challenges? Again, we have the promise and the impact, but really here are the challenges that we have to kind of confront. Again, the first is complexity, right? Going from a minimal cell to the E. coli, it's a 10x increase in genes. But here is where I think sort of the DUF effort kind of comes in. And this is basically saying that, we have the genome, we can sequence everything, and we can make pretty reasonable guesses about what most of those proteins are doing. But still, 35% of proteins plus or minus have unannotated protein function. Again, we can guess, we can make inferences, but we don't have sort of a credible ground truth that sort of substantiates a lot of these hypotheses. Also, there's other issues of having proteins having moonlighting functions and other kinds of functions beyond what we currently know as well. So this is kind of bucketed into this challenge of what we call sort of sparse data. And of course, there's issues of noise and stochasticity that have to be confronted as well to sort of fully realize, again, the value and the potential impact of these whole cell models. And so, I think there's some really interesting opportunities in the ability to basically sort of, harmonize new data, for example, some data that comes from this DUF program, but also new techniques, right? And so you can think about interesting advances on the modeling and compute side, and harmonizing those with interesting advances and sort of the data acquisition side that would come from the DUF program. And so, I think taken together, if we had this virtuous cycle where the model can help us make predictions about experiments, and that data from those experiments can help us improve the model, we have this virtuous cycle where you can kind of improve both of those. And I think that's really kind of an important mechanism to advance whole cell models and really port them to other fields and advance them to other stakeholders that might be interesting. And so just on the experimental side, I think there's a lot of exciting advances that are coming across the wire, so to speak, right? And so I think those are well characterized in this program and other programs. On the computational side, we think there's some really interesting advances as well that could basically use those data more effectively in the context of whole cell models. And so I think an exciting advance that's been explored by us and others is the idea of sort of physics informed neural networks, right? So basically, this is the idea that we can sort of solve high dimensional equations, sort of systems of PDEs very efficiently, right, with the right insight. And so we can basically take a little bit of data and make important inferences and powerful forecasts about how dynamics of systems will evolve in time. So I think this is a way we can take some of the data from Duff and really kind of amplify the impact in one context. Another innovation that I think is interesting when we think about bringing to bear compute with data in the context of whole cell models is liquid neural networks. And so the idea here basically is to think about, going away from just sort of, simple, fully connected embedded layers to more complicated interconnectivity between those layers. And so we can have basically higher expressivity, we can have feedback and nonlinearity between each of the neurons and these different layers. And so in doing so, we can basically create a more advanced understanding of system dynamics with a lot fewer neurons. And so in various contexts, this could be, orders of magnitude fewer neurons that are connected in a more meaningful, expressive way, but basically we can get some really exciting and accelerated compute output from simpler neural networks. So I think that's another exciting tool that, when leveraged with things like pins, with leverage with new data from the Duff program, could really be ported over and really could transform how we think about the trajectory for whole cell models. And so that's really kind of my conclusion for you today, and the message I want to leave with you is we think whole cell models are a really cool way to think about understanding biology and performing in silico experiments. And these models could have advantages from recent advances kind of on the compute side, but most importantly, they could have really impactful and exciting advances by leveraging a lot of the data from the techniques from the Duff effort. And so with that, I want to conclude. I want to thank again Renee for inviting me, and thanks to you for listening. And I hope to see you again in the near future. Thanks and goodbye.
Okay, welcome back, everyone. Thank you for sticking with us for our small group discussion. So online, you are going to be randomly divided into 8 small breakout rooms. And in the room here, we're going to have 4 groups of 4. So in your small group, you need to choose a speaker and a note taker and discuss the future of gene function annotation. So question 1, if money and expertise were no object, what's your dream for discovering unknown gene function? 2, where do you want your field to be in 20 years? And what do you envision needs to happen in order to make that a reality? And then during the out briefs, your speaker will summarize your group discussion and answer those questions. And lastly, the note taker can email the notes to me, renee.chavir@darpa.mil. And we will come back together in 45 minutes. Right, 40 minutes. Okay. All right. Thank you. Okay, at 4:45. Excuse me, 3:45. Okay, so in our first table over here, I have I'm just going to say your last names: Agman, Charlotte Powers, Chouard, and De Crécy. The one next is Di Paulis, Fader, Falston, and Gill. And you have the whiteboard and the paper up there to use. Oh, okay. Thank you for that update. All right, on our next table up front, oh, did I say Gill? Okay, good. Okay, thank you. On our next table here up front on the right is Hardy, Knight, Long, and Magyar. I really apologize if I'm pronouncing your names wrong. And I'm sorry, Dr. Wong, you can join this group right here. Yeah, that one. And then there should be 4 remaining, and you have the last table up front. Hello, everyone. I want to say thank you for all the wonderful discussions. We are going to finish up our small groups. So if you could tie up your conversations in the next 1 or 2 minutes and finalize what your speaker is going to say during the outbriefs, we are going to outbrief our small groups here in the next 2 minutes. Note takers, go ahead and email me your notes if you would like them to be projected. I can do that. Oh, yeah. Okay. Yeah, you don't have to put your notes up if you don't want to. Yeah, but please still send me your notes. Welcome back, everyone. Go ahead and finalize your notes online. We're going to start the outbriefs shortly. We had some really amazing discussions here in the room. I hope that your breakout sessions online were just as good. Just your speakers can do their presentations in just a moment. We're going to go ahead and start with the in-person first, if that's okay, and then we'll switch over to the online. Group 1, if you're ready. Sure. Oh, yes, she does. I don't believe that. All right. Do you want to put anything on the white? No? Okay, cool. No problem. All right. All right, there you go.
Okay. So group 1, we spent a lot of the entire time talking about just the challenges of data sharing, data curation, trustworthiness of database. That also the fact that a lot of existing databases, as soon as the funding runs out, they're no longer maintained, no longer valid. So just so much time and churn, both on the experimentalist side and on modeler side, is trying to get the data. And if you have multiple databases that you want to integrate into, they often will disagree on annotations. So for our question 1, the infinite money dream, we want to coordinate these efforts. There needs to be funding for a reliable database. We actually called it like a data federation. It might not be in one place, but we need more of a distributed network in which different data can be posted, communicated. Our dream is that if an experimentalist comes in, has a new annotation or some new knowledge, update the database, it'll propagate to everyone else's database. And if you're running models, your model will have now been updated and is improved. We want this to happen faster, and the turnaround both on the experimentalist and the modeler side. So it's just turnaround. Yeah, like UniProt, we actually spent a lot of time talking about UniProt, and people in the group were quite pleased with it, but I think not quite there. Maybe we can, if the chat mentioned UniProt, so if we want to bring up something about that, we could. The field in 20 years is one in which the knowledge becomes available instantly upon it being retrieved. You post your data, everyone can now access it, and we don't have to go digging deep and finding synonyms and making all these mappings that really take up a lot of time. A different kind of science becomes possible when turnaround time is fast. And I know this from the modeling side. If I can run a model and get the results before I get up and have lunch, and before the next day, and it's still fresh in my mind, I can, within that same session, debug and iterate on the model. If I have to wait, if there's a gap there in which I have to wait for someone to validate and give me the correct annotations, and now it's weeks or months or years before I come back and interpret my results, I'm not doing science as efficiently. So just the turnaround time related to annotation and curation, I think, is the main focus here. We had a few different ideas. I think it requires a conversation to really figure out how to do that well. There's been a lot of efforts to do it, UniProt being one of them. And I'm not sure if we really have a solution. We talked about the internet protocol kind of here in DARPA. I think it is even more of a relevant topic. That IPTCP was invented decades ago, and it's mostly been unchanged. Meanwhile, the internet has undergone many revolutions. The internet's changed enormously. The protocol is the same. If we can come up with a protocol for data sharing, and so we can boot up servers with databases and they communicate seamlessly, I think we could overcome some of these challenges. All right, good time to pause. And did I cover everything? Is that good? All right. Any questions?
All right. Thank you very much. Group 2, you're up.
All right. Hello, everyone. So our group discussion really focused on phenotype. It's one thing just to know what's there, but we need to know, what the expression of a cell, how it's reacting to any particular environment. And so one of the things we really talked about is we kind of just need more of everything. So if we have infinite money and expertise, one of the things that we were thinking about was getting, more genomes, transcriptomes, proteomes, metabolomes. So that way, we can kind of get a more complete picture of how cells will be reacting. And this isn't just for bacteria, but also for eukaryotes, because again, we have to consider that the systems and pathways that are found in prokaryotes are going to be very vastly different in eukaryotes. And so if we have unlimited resources, just getting a complete picture of what's happening at that level will be great. We also we're thinking about, looking at different environmental conditions, because that might affect, various aspects of the expression, but also of the behavior of the proteins. Some of the discussions we had were looking at how environmental factors might affect protein folding. And so we were thinking about if we could have expertise kind of look into modeling and developing models that incorporate, physical and biochemical factors that might affect protein structure to improve protein modeling. How's pH, salinity, temperature, all these other things affect protein structure and i.e. function, would also be useful expertise to have. In the future, in 20 years, what we decided would be really useful for the field would be to have like single cell proteomic potential. So we can actually see what the entire proteome is like, see what proteins are there, what are expressed, what are not expressed, in these different conditions, because the transcriptomes, just because there's RNA there, it might not be behaving the way we think. And post-translation modification definitely is a real factor to consider in these conditions. And so that's really kind of the brief summary of what we did. If you have any questions, I'll try to answer them. And if you had group 2, if you have anything to add, by all means.
All right. Thank you very much. Group 3.
So our group had the bold suggestion to basically just throw away all data that's ever been made. Thanks, Tom. And then create so much more data, very similar to what the group 2 had just said. So, genomics, transcriptomics, proteomics, metabolomics, and using just sort of state-of-the-art tools to get this data, but most importantly, linking it to real-world metadata. So the ability to collect environmental samples and data from interesting places around the world is something that's really interesting, but you need to be able to basically connect what's happening in that environment to what's happening with the transcriptome to have that be meaningful in any way, shape, or form. But even more important, that sort of data is not useful without standardization. And so basically figuring out ways to standardize both on phenotypes, but also how you're sort of documenting those phenotypes and making it so that basically it's digestible easily by computational systems was something that we thought was really important. And then for question 2, really we were imagining a world where biotechnology and biology is like where the semiconductor industry is, where you can go and use a design tool to create complex biological function. If you want to have an organism that produces, a particular molecule at a particular temperature and using some sort of like complex feedstock, but then leave behind some other high value product from that feedstock. So just really sort of map out like eat this, not that, do it at this temperature. Something like that would be just so powerful. And then to get there, it's really about a lot of this knowledge management, high throughput data generation, and the multiomics and modeling. So just improving in all those areas, sort of like where we described the path for question 1. So any questions? Happy to take it. Otherwise, thanks.
All right. Thank you so much. Our last group.
So there are a couple different themes that emerged from our discussion. One of the first ones was that in other fields like physics, there are some problems that really could only be tackled by building something like the Large Hadron Collider, which took cooperation from different countries and pouring a lot of resources into building this giant machine. So we talked a bit about this. Could something analogous be built for for biology in the sense of having like a center or like maybe like a national cloud lab that a ton of resources get poured into to set it up to be able to run experiments at a really like large scale, much larger than any individual company or group that has done thus far. And you'd need that sort of expenditure to. We've seen existing cloud labs have had problems with their business models, and some of them have failed. But if that was like propped up by the weight of government, you could get something that that could potentially work. We identified there's sort of this tension between, on the one hand, there's like different ways you could go about getting gold standard or data that works with computation. On the one hand, you could try and collect all that data in the same environment so that you have some degree of guarantee that the conditions you're collecting it under are equivalent. And that's something that a Large Hadron Collider type national cloud lab, that would sort of, go away towards solving that problem. The other way you could tackle it is if you made better standards and ways of collecting the data and distributed them, widely enough that you could make the data being generated by smaller entities, smaller organizations, more comparable and computationally tractable. And to some extent, that is happening organically right now. Within all the different subdomains in biology and different things, people are coming up with, ontologies, different ontologies. Some of those are redundant and, they overlap to a degree, but that overlap is actually kind of useful. So another vision for the future that's not completely orthogonal, potentially complementary to a Large Hadron Collider is that you have, we're across science, we're starting to come up with more standards for collecting, collecting metadata and organizing it. At the same time, we have development of, AI, and we're seeing the potential for better, artificial agents that could potentially help to, take that disparate data and try to reconcile it, put it into a common picture and present it for a human to read in a more natural language format. Because speaking as someone with a lot of experience in standards, 99% of people, they're not going to look at the data in the standard format that you come up with to make it digestible for the computers. So, even in a world where our data is, perfectly standardized, we're going to need the systems in place, like some of the previous speakers were talking about, to help, federate that data or organize, basically to do this, help us do the synthesis part of it in a much, faster, more automated fashion than what's currently accomplished, which takes, which takes individual scientists reading a lot of papers and thinking about it for months and months to come up with, summaries of the things that they found. So anyway, those were two of the major themes that we talked about of, like in terms of vision for the future is, having something like Large Hadron Collider for biology, where people could, that you have a ton of national resources invested in it, and people can send in experiments to be done at it. And there's, this additional guarantee that things are being collected under equivalent conditions, and then putting in better systems in place for synthesizing the, the knowledge that we've collected thus far, and will probably continue to collect in the more, individual entity fashion, because that's still going to happen on some level, because that's what we do. So that anyway, that's broad strokes what we talked about. Did I misrepresent anything? Yeah. Okay, great.
Great, thank you so much. Thank you. This was wonderful. I hope that it was helpful. All right, now we're going to switch to our online groups. I know there was a little bit of confusion with the small groups, and I apologize for that. So I believe we ended up with 3 groups altogether. That's not correct, just let me know. But I think if we could start with room 4. And if you want to share your screen for notes, you can. So just go ahead, the speaker, unmute yourself and go ahead and present. All right, maybe there wasn't a group 4.
We didn't know our room numbers, so maybe there is a group 4. We just didn't know where we were.
Who knows who is 4 or 3?
We don't know. We don't know our numbers.
Okay, how about this? Someone just go ahead and go.
I think we were room 5. Hello, this is Nesli Hantash from Berkeley Lab. So I'll just jump in and just start reporting back. Sorry, I don't really have any slides or screen share to do. Just to keep things a little bit on track, I'll just talk about the things that is not being talked about by the other groups. Coming to the question 1 of unlimited money and resources. We did talk about similar things like making, just extensive libraries, better wet lab confirmations for the functions discovered, ease of use things like one push button and we get all the information that we ever need, good confidence and, like reliable sources. So that seems to be a common theme, I think, for this particular question for close to everybody's heart. Besides that, there were a couple of things that was not really mentioned, so I would like to call them out. One of it is that to get there, we will probably need better assays and methodologies for validation. Just currently the state of the art is not really actually serving the full needs of the community. So there will be, just some investment needed to facilitate that. And another one is that there are multiple disciplines trying to tackle this question of discovery of function together, ranging from experimentalists to computational biologists. And the scientists do not necessarily share a common language. What enough information for one might not be enough information for the other one. Maybe, Ido, if you could be so kind to share that article that you shared with us within the group to everyone so that they could see. This is a thing that we also need to maybe talk a little bit about more, how we can actually find the common points that everybody agrees what kind of information that we need to collect and analyze to get where we want to get. When it comes to the question of where we would like to be in 20 years, I think the common theme was that everybody wants to have something easy to do and meaningful when it comes to discovery. How to get there could be, could be very wildly different. We discussed, just 20 years ago, most of us were not really doing what we were doing nowadays. Sequencing technologies, for example, they were drastically different than today. So we discussed about how that could be different. Just maybe we can actually have infrastructure that, just does many analysis all at once on the fly, including the analytics, so that, just it really becomes a one push button or a turnkey technology. There's also a likelihood that maybe sequencing will not be as important as it is today now. There might be a complete revolution that it is needed that, makes us move away from sequencing and start doing other things or other targets or focusing on, just more processes than the sequence data itself. And it was mentioned that, just if we actually do not do half of the things that we are doing right now, maybe that's an achievement because it is not needs to be done anymore. So I thought that was a very interesting point of view to have. I think, just that more or less summarizes our discussion.
Wonderful. Thank you so much. All right, next.
Again, I don't know which group I'm in, but I want to say like my group member is Kristen, Arena, and Sandra, and myself. So I am a research scientist from Joint Genome Institute, and then we discussed in our break room, and there's 3 points. Basically, we agreed. The first one is figure out the details of the pathways from different angle. So that need like a lot of data generation and assays. Second one is like want to know so current approach work on the comprehensive gene functions. Still some probes are still expensive, so we need to like it's nice to have some approach which can be cheaper to generate the data and then share the big data and then do data mining. And then the last one is figure out the cis regulatory element, the function of the cis regulatory element, and then develop a approach which can do high throughput functional validation for those cis regulatory elements. We combined both questions together. Like, I mean, that's our dream work. Also, like, we hope that will be clarified in 20 years. Yeah, thank you. I don't have other like notes or sharing, and because Kristen take notes, and it's just based on my memory to speak out. Thank you, everybody.
That was perfect. Thank you so much. And next group, please. Is there another small group online to share? Okay, I'm going to take that as a no. Note takers, if you would please email your notes to me. Okay, all right. All right, well then that went a lot faster than I was expecting. But I really appreciate all the brainstorming and the ideas. I hope that it was helpful. All right, we are going to move on to the Discovering Unknome Function Advanced Research Concept Answer session. So throughout the workshop today, participants were emailing their questions and submitting them here in person. So I'm going to go over some of those answers. All right, first question, how can I apply to the DEF-ARC opportunity? All right, so we encourage you to visit sam.gov or darpa.mil/ARC, ARC, and read the Exploration Announcement Master Solicitation for Advanced Research Concepts. And that number is DARPA, DARPA-EA-24-01. And it is dated October 27th, 2023. And then the specific DUF opportunity is DARPA EA 2401-02, Discovering Unknown Function, DEF-ARC opportunity. And if you have additional questions or need assistance, please email duff@darpa.mil. All right, next question. For studies that may require significant experimental work, are the proposers expected to find funding beyond the $10,000 available for supplies and reagents? Per page 1 of DARPA EA 240102, DEF-ARC opportunity, proposed costs are limited to $10,000 or less for materials, equipment, and other direct costs, ODC. Any proposed ODC beyond this $10,000 limit will be the responsibility of the proposing organization via cost sharing. Next, can you elaborate more on the funding allocation? Specifically, can more than one individual work on this project? Per page 1 of DARPA EA 240102, the DEF-ARC opportunity, while multiple researchers from the same organization may be proposed, the aggregate level of effort for a proposed research concept must be equivalent to one full-time equivalent FTE and 12 months. DARPA expects that the individuals working on the proposed idea primarily focus on the effort for the entire period of performance to the maximum extent practical. Only minimum variation to this requirement will be accepted. Will the presentations be made available? If we are able to obtain the necessary approvals, we may decide to post any relevant materials to darpa.mil/ARC under the resources section on the right-hand side of the page. Next question. What technical objectives should the abstract address? Per page 2 of DARPA EA 240102, the two technical objectives are 1, generation of high-quality data sets to hypothesize or predict gene function, and 2, experimental validation of these hypothesized or predicted gene function. You can do one or the other or both. Next question. is studying protein-to-protein interactions within the scope of this ARC opportunity? Yes. However, one of the proteins must be tied back to a gene with little or no annotation. And that's it. Okay. That is all I have for you today. I just want to extend my extreme gratitude to our speakers today for their efforts and preparing their slides and talking, and for all those who came in person and were online. And I hope that some brainstorming happened and that everyone learns something. So if you have any questions, feel free to reach out to duff@darpa.mil. And thank you very much and have a nice rest of your day.